Greetings and welcome to Fibroid Awareness Month 2026. This is our third PCORI-funded convening, and we are so excited that you are here to join us. The Fibroid Foundation is in its 13th year, and our mission has always been to bring advocacy to the fibroid space. Over the years, that mission has expanded to include support and advocacy across the menstrual spectrum in and through menopause. We are looking forward to this session. The Patient-Centered Outcomes Research Institute, also known as PCORI, compares healthcare approaches to determine what works best for whom and under what circumstances. PCORI's mission is patient-centered, which is key, and that means that the community's input directly informs what gets studied next. This convening is a targeted effort to share findings to help shape future research priorities. Today, we will hear from Dr. Mazhar Adli and nutritionist Lenice Brothers. The focus of this session, Expanding the Fibroid Treatment Landscape, brings together these experts in the fibroid field, focusing on innovations, therapeutics, and holistic options. Welcome, Dr. Adli and Lenice Brothers. We are so pleased that you are here to join us today.
So in the next few minutes, I would like to share some of the efforts in the lab going on, and some of these efforts are now we're hoping to bring some therapeutics for fibroid treatments. As our audience know, the fibroid tumors are the most common tumors in humans, more common than any other type of tumors. 70 to 80% of women will have them, and about 30% require some sort of medical treatment, actually. And there is no approved drug treatment strategy for these tumors. At the genetic level, the genetics of these tumors are relatively simple compared to most cancers. About 77% of tumors are driven by this point mutation in this gene called MET12. And there are several other genes that are also mutated. And the interesting thing is that each of these mutations are mutually exclusive, meaning that tumors only need to have one of these mutations. So since the most common mutation is in this MET12, it requires special attention. How can we model this MET12 mutation? And after modeling, can we find therapeutics targets to this one? I'm saying the modeling because there was no cellular models to study this disease. I think this has hampered the progress in this field. In cancer field, as someone working in the cancer cell lines, we have thousands and thousands of cell lines for cancers. But yet, for this most common tumor, there was not a single cell line that people could use in the laboratory to study what is the function of these mutations. So this lack of cell models, I think, has been a significant roadblock in the therapeutic development, in the even understanding the molecular biology of these tumors. So we took this challenge to study, to identify this cell model that we can study this. The way we did it, we used CRISPR gene editing technology, which is a really cool technology that you can go into the genomic region and change any nucleotide to any other nucleotide that you want. So we took normal cells and we said, let's engineer that mutation that we see most often in patients, which is in this MET12 gene. And let's see if this normal cell turn into a tumor-like cells then. So we did that. Of course, I'm going over this 5-year work very rapidly, but we show that this engineered cells, that when we engineer that point mutation that we see in human, they show much higher 3D and in vivo growth, extracellular matrix production that we see exactly in human patients. Gross pathology were very similar. The gene expression program, the metabolomics were very similar to human fibroid tumors. So this was very encouraging for us. We published this, and now we are sharing the cell lines with the community. But most importantly, now we have a model that we can actually now start seeing what can work on this model, what can actually revert this aberrant phenotype back to normality. So we took this challenge. We said, can we find drugs that will selectively kill these mutant cells and will let us to identify this potentially clinically relevant targets? So we designed this assay where we are growing these mutant cells that we have engineered together with the normal cells in a 50-50 kind of ratio, and we start treating them with drugs. And so far we have screened 30,000 drugs. And I'm happy to share that we have identified several drug candidates that seems to be selectively killing the mutant cells. We already did a secondary screening for these drugs. And now we wanted to actually show that it works. It shrinks this mini kind of fibroids that we can grow now in the lab. So in the control cells, these fibroids, when we grow them in 3D spheres, kind of organoid condition, they grow to have a very significant size. And when we add the drug, they kind of shrink. And this is that data showing here. We also did a co-culture experiment. This is actually the mutant and wild type cells are grown together. As you can see, after 4 days even, the mutant cells kind of take over. So the whole spheroid, the whole mini fibroid becomes green. But when we add drug, these green cells are dying, the mutant cells are dying, when the whole thing becomes red. To finally show that this actually is relevant to human, we have this system that we can culture these tiny slices of actual patient samples. So these are actual patient tumors. We can culture them in the lab for up to a week, and we start treating them with this drug. But the challenge here is that we want to show that we selectively kill mutant cells, but there is no assay to actually see and quantify that you are selectively killing certain cell types in a tissue. Because when we do this assay, we cannot differentiate which one died. Was it the wild type cell? Was it the mutant? So we set out to actually design a new assay, which is a next generation sequencing based assay. We can actually read the DNA in these samples, and we say, did mutant reads kind of went down with the treatment? And this is what we see, that selectively we see that the mutant reads are going down when we treat these patient tumors. So we are very excited about this. Actually, we are now we set up a company, and we want to bring this to therapeutic to the clinic. We have a name now. We just recently incorporated. The name is Rosa Therapeutics, and we are hoping to bring this molecule that we have, and also we have several other candidate molecules for treatment of fibroids. So the company is targeting both uterine fibroids and also PCOS, which is now called PMOS. We start with the human genetics, develop model systems for them, perform high throughput screening, chemical screening, CRISPR screening, so that our probability of success becomes very high, and hopefully we target these two. And the company, I am one of the founders, but I have two amazing people who are helping me on this journey. Cosmas has a lot of experience with the company formations, and he was a CEO of several companies and exited. Elnur is another friend of mine who is also a physician scientist at Northwestern University. This is a new journey for me, but I'm very excited, and I'm thankful for your invitation. It's a pleasure to be here. Thank you.
Thank you, Dr. Adli. That's fascinating, and I'm sure that our listeners will find it fascinating as well. I know we will have a Q&A session later, but I want to ask a couple clarifying questions before we move on to our next speaker. Can you define for someone who is not a clinician what Med 12 is?
Yes, absolutely. Sorry, I should have clarified. So in our genome, we have 20,000 genes, and MED12 is one of these genes. Normally, it is involved in the transcription of other genes. So MED12 is part of this complex called mediator complex, and it has a 26 subunit complex. It's a very large complex. MET12 is one of these subunits, and it is involved in the kinase module of this one, which means that it brings phosphorylation to other proteins. So it is required for the normal expression of many other genes. When this mutation happens, we show that the gene expression program changes in the cells, and their metabolism is also changes. And we still exactly don't know at the molecular level why this particular mutation is affecting its function, because it's not a loss of function completely. Because if it was a loss of function, we could lose MET12 completely with several other mutations. But we keep seeing the same exact mutation in MET12, which typically indicates that this is a kind of a gain of function. But all the research so far we have identified is that it is a gain of function because certain genes go up as well. But the kinase function is diminished in a way. So the whole complex is less functional, but it's not completely unfunctional in a way.
Okay, that's helpful. And what's kinase?
Uh, so kinases, we use this term as scientists, any protein that puts a phosphate group on another protein. So usually the cell signaling, in the way the signaling mechanism happens in the cell, phosphorylation and several other chemical modification happen on proteins. And this is one of the critical modification that tells this next protein that now it's your turn. Now you go do this thing. So when that phosphorylation doesn't happen, in a way, that kind of signaling cascade doesn't go down. So that's one of the functions of this module.
That's very helpful. And last question, because I snuck one in there on you. Uh, I'm very curious about cellular models that you mentioned, and I wondered if you had any thoughts on how or what would be required for us to get a number of cellular models that really is reflective of the magnitude of this health concern so that the research can move forward more expeditiously.
Yeah. So we developed this model. I'm currently sharing for research purposes with other researchers as well, and we just set an MTA with them, and I am sharing these cell lines with them. But mm, this is one of the models, to be honest, meaning that it's just one model because there's no other model, but we need to engineer additional models. First of all, we just modeled one of the mutations, but in this gene, we have several other mutations that I think we should model them, create the models for them as well. Secondly, although this gene, this MET12 is the most commonly mutated mutant, we have several other genes also that are mutated. I think we should create the models for them too. And lastly, I think we should now compare these models to identify whether there is a common pathway, a common program, aberrant program among all these models that we can target. Like right now, although we are very excited about this molecule that we have identified, but currently I don't know if this molecule is going to work for these other mutations or not. I'm hoping it will work, but we don't know. And this requires generating these additional model systems. I think that's one of the key things that as a field we need to focus on engineering new model systems.
Thank you for that. And thank you for, sharing what we don't know, because that helps us move closer to finding the research funding to uncover those unknowns. Very informative, Dr. Adley. Thank you so much. Looking
Thank you.
forward to learning more during our Q&A.
Thank you.
Um,
You're
at
welcome.
this time, I'd like to introduce Lanice Brothers. Lanice is a nutritional therapist and yoga teacher specializing in helping women with issues with their menstrual cycle, painful periods, and balancing hormones. Lanice is based in the UK and has been a longtime alliance partner of the Fibroid Foundation, and we're always so thrilled for her partnership and the knowledge that she brings to our community. Lanice believes in the power of food as medicine, something to be enjoyed and celebrated, not feared. She's a member of the British Association of Nutritional Therapists, a regulatory body that oversees nutritional therapists in the UK, and the Complementary and Natural Health Council, a UK registry of complementary health practitioners who have met UK standards. Lanice has helped hundreds of women improve their menstrual and hormone health through her private practice and group programs. I am one of those women, and I am super thrilled to introduce Lanice Brothers. Lanice, welcome.
Hi, thank you so much for having me here. So what I am going to talk through in my session is talking more about the hormonal side of fibroids, talking about what you can do from a nutrition perspective and also a lifestyle perspective that I think is really important to take into consideration. With fibroids, they can be very disempowering. We There's a lot of information that we don't know. So as a patient, you go to your GP or your doctor, and they say, well, we don't know why they grow. We don't know where they come from. We don't know why they start. But what we do know, well, one of the things we know is that estrogen or estrogen in the US is the fuel for fibroid growth. And that's really what I'm going to focus on today, the role of estrogen. We know that estrogen causes fibroids to grow. Estrogen stimulates fibroids because fibroids contain more estrogen receptors than normal uterine muscle tissue. And so fibroids are very sensitive to estrogen. And so when we think about estrogen or estrogen, we have to think how we are going to balance estrogen in the body. And we think about it in three ways. We think about the estrogen that we're producing in our ovaries and also in our adipose or fat tissue. We think about the balance between estrogen and progesterone. And then we also think about how we're supporting our body in the detoxification of estrogen. And we know that happens in the gut and that happens in the liver. And from a patient perspective, looking at the management of fibroids this way can be very empowering because a lot of this can be done through nutrition, through supplements, and through lifestyle management, as I mentioned earlier. So if we think about first what we can do through nutrition, so looking at how we're managing what we call the estrogen load, we look at firstly what we're adding in through food to support the liver. And I know that a lot of doctors might say, well, the liver detoxifies itself, leave it alone. But there's a lot of evidence that tells us that when we add in certain nutritional compounds, certain nutrients, we give our liver a helping hand. And this is really important because we want to make sure we don't have excess estrogen because as I mentioned, that excess estrogen is fuel for fibroid growth. So thinking firstly about what we're adding in, really looking at for the first phase of liver detoxification. So liver detoxification happens in three phases. In the first phase, we're looking at adding in nutrients like magnesium, which you can get from nuts, from seeds, from dark leafy greens, looking at a nutrient like selenium. Selenium you can get from meat, you can get from Brazil nuts. I think a lot of people know the connection between Brazil nuts and selenium. And selenium is really interesting as well because it's really important for supporting the thyroid, which is this gland at the base of the throat. And thyroid management, just to go on a slight tangent, is really important for fibroid management because when the thyroid is really impacted by estrogen imbalance. And so when we manage the thyroid, so making sure we're, in this case, adding in nutrients like selenium, like zinc, like iodine, like iron that help the thyroid produce the hormones that it needs to produce. It's another route to helping to manage fibroids. So then going back to other nutrients that we can add in, and I'm just gonna rattle through these, but if you want to know more about these specific nutrients, of course, there's a wealth of information on the internet. There's also a lot of information in my book, You Can Have a Better Period. So zinc is a fantastic one that you can get from pumpkin seeds. Vitamin C, citrus is a fantastic source of vitamin C, vitamin B6, folate, vitamin A. Then we also look at the next phase of liver support, phase two. So this is where we start to look at cruciferous vegetables. And these are vegetables like kale, like broccoli, like cauliflower, Brussels sprouts, kohlrabi. And these are really valuable because they contain a compound called sulforaphane. And this compound is very, very important for helping the liver break down the estrogen that the body has either made or absorbed. We're also looking at allium, so onions, garlic, spring onions, carrots, ginger. All of these contain nutrients that are really supportive for liver. Then we look at the phase three, which is excretion. So this is where having a bowel movement every single day is really important because this is how one of the ways that estrogen is cleared from the body. And so when it's effectively cleared from the body, we make sure that it isn't, we don't have a situation where we have increased levels of an enzyme called beta glucuronidase. And this is an enzyme that can reverse the process of breaking down estrogen and have it reactivated back into circulation, exacerbating that potential excess of estrogen that then fuels fibroid growth. So the first part of this nutrition section is really thinking about how we're supporting the liver. And if you take anything from this, it's really eat more green vegetables, eat some carrots, think about what you're adding in rather than what you're taking out. Then we also have to think about how we support the gut. So in our gut, so we have I think a lot of people know about the gut microbiome. So this family of specifically bacteria in this case that helps our body break down that helps support our immune system. But in within the microbiome, we have something called the estrobolome. And this is a specific family of bacteria that helps the gut break down estrogen. And so when we add in foods like fiber and different types of fiber, like soluble fiber, insoluble fiber, this is another way that we give our body a helping hand to manage estrogen. So soluble fiber, I'm sure a lot of you are eating oats for breakfast. That's a fantastic way that you're supporting your gut health and supporting the body in breaking down estrogen or estrogen. Other sources of soluble fiber, green bananas, so unripe bananas. And some of you might think, okay, that's not very appetizing, but they're really helpful. Psyllium husk, citrus again, beans, lentil, chickpeas. Then we have insoluble fiber, and we might think of this more as like roughage. So thinking about the peels, edible peels of fruit and vegetables, brown rice, wholemeal bread, nuts, seeds, even something like popcorn can be really beneficial. So we've talked about the liver, we've talked about the gut, we've talked about making sure that we have a bowel movement every day. All of this is really helpful for moving estrogen through the body. Then we look at something like xenoestrogen. So xenoestrogens are basically synthetic forms of estrogen. And we absorb these through the skin. We also breathe these in. So something we think about personal care, so skincare, beauty products, makeup, even something as simple like Febreze. We breathe that in, and the body treats that like a synthetic form of estrogen. And when I work with women with fibroids, I always have them do an audit of the different products that they're using because this is a shift that you can make and it helps you feel quite proactive. Um, to moving away from these kind of common everyday products, something like a Tide for cleaning your clothes, which have a lot of these synthetic estrogens or xenoestrogens, to more natural and organic products that won't increase the estrogen load. And so, if this is something that resonates with you, it's worth going through your skincare products, beauty products, personal care products, cleaning products, and just thinking about everything that you're using and where you can make shifts. The Environmental Working Group have a really great app and you can look at what's in all of the products that you use, and they make suggestions for switches that you can make. So that's really powerful from a lifestyle perspective, where you're actively managing estrogen um, absorption in the body. Last point here, um, vitamin D. Vitamin D is so important when we think about fibroids. We talk a lot about estrogen and estrogen, but we also have to consider the connection between vitamin D and fibroid treatment. So, vitamin D receptors are actually decreased in fibroid cells. But what we see in the research is that vitamin D treatment actually decreased fibroid proliferation and signaling. And there's actually an inverse correlation between serum vitamin D levels and the presence of fibroids. So what I would encourage is to go to your GP and have your vitamin D levels checked. And we know that fibroids are incredibly common in Black women and women of African ancestry. We also know that typically there are lower vitamin D levels in the same population. We also absorb less. It takes us longer to absorb vitamin D from the sun. So this is something, again, you can be proactive with. Go to your doctor, get your vitamin D levels checked, and then work with a practitioner to make sure that you're supplementing appropriately. And we're in the summer months, so it's easy to get vitamin D when you step outside on a sunny day like today in London. Thank you so much.
Thank you, Lanice. That was incredibly informative. Wow. I have never heard anyone discuss estrogen absorption through the gut and name it. That's really fascinating. Um, how do you have any recommendations for how our listeners could um, better understand that dynamic of the gut estrogen absorption? I just find that to be huge.
Yeah, so I think the first step is really understanding the gut itself. So when I talk about the estrobolome or the estrobolome, that's just a subset of everything that's going on within the wider gut microbiome. And we're in an exciting era where we're learning more and more about gut health. And there's a huge amount of research, certainly in the UK, around gut health. Um, we're still learning more about the estrobolome, but what we do know is that it's hugely important for making sure that our estrogen levels are balanced and that we're not seeing estrogen recirculation happening in the body. And as I said, that's very, very important when it comes to fibroids.
Wow, that's huge. And I did not mention your podcast, Period Story, which I think is fascinating, where what's the first question you ask everyone when you start Period Story?
So I say, tell us the story of your first period.
And that question is so incredibly unifying. And for me, when I was on the podcast, it was telling because it really dictated my period journey and indicated some things that I hadn't thought about in a very long time. So I encourage everyone to visit Period Story podcast and listen because Lanice has interviewed over the years many, many, many people and many people with fibroids. And it's fascinating. Um, so as we conclude our presentations, just so much really incredible information that you both have shared. And I'd like to ask a few additional questions of you both to round out our really just incredibly informative time. One of the questions that always comes to my mind is, and I'll come to Dr. Adli with this. Are there any plans to study women without fibroids? Can we learn from that subset?
Yes, absolutely. I mean, one of the things in the scientifically that we can do is to study the genetics of people with fibroids versus the genetics of people without fibroids. And this kind of research is incredibly helpful and useful to really identify which part of our DNA can be associated with the development of these fibroids. We have investigated 50,000 people with fibroids and more than 100,000 people without fibroids and compare their genetics. This study has been done usually by other people called genome-wide association studies. So we, for example, a few years ago, we collected all this published research and find what can we learn from this one. And then we found that there are like hundreds of locations in our genetic information that are associated with fibroids, but this is an association. It's not really causal, meaning that if you have, because between any two people, there are 10,000 nucleotides or letters that are different between two people. And in our genome, we have 3 billion letters. And between any two person, about 10,000 of them are different. And some of these differences are not necessarily mutations, and most of them are not mutation. They are just natural variations in our DNA. And even these natural variations can be associated with it. When we study compared to normal people, there are certain parts in our DNA that are associated. And what we can do now, or what can be done, should be done now, is what is the meaning of these small associations? What are the genes that are regulated by these small variations? Because this, as the name suggests, they are very small, meaning that their impact on the gene expression is not like completely all or none. If that target gene was expressed, let's say at 100% unit, 100 unit, now with this variation, it is expressed at 80% or maybe 110%. So the variation is subtle, but collectively they may associate with the fibroids. So as you said, it is incredibly useful to compare it to normal people and see what is the molecular mechanism.
Any plans to write a paper on what you just described if you haven't already?
We have published one paper. We have another paper coming on actually. So we published the initial kind of descriptive nature of this work. Usually in this genome-wide association studies called GWAS, the challenge is not to identify which part of the DNA is associated with the disease. The challenge is really to study now this part that we have identified to say, now what are the genes regulated by this? In which cell types? Because the biology is extremely complex. We
Mm,
have more than 200 different cell types in our body. And the cell types that are associated with the fibroids, we know that the causal mutation that we typically see, like MAT12 mutations, they are in the smooth muscle cells.
mhm.
So it happens in the stem cells of the smooth muscles, and this leads to the fibroid formation. But we know that the immune system is very important in these things. As Lenise described, several hormones come together and impact the proliferation of not only these mutant cells, but also the proliferation of fibroblasts, for example, around these mutant cells, or the non-mutant smooth muscle cells. So these genetic variations then become a critical determinant, of other cell types. Studying them, I think, coming back to this generating kind of faithful model systems for these diseases is going to be extremely important to kind of disentangle all these different impacts that come together to cause tumors.
That's such incredibly helpful information that you're sharing. Thank you for that.
Okay.
Uh, Lanice, Uh, I know we talk a lot about fibroids and reproductive age women, but I know that you also work with women who are perimenopausal and transitioning in and through menopause. And there are some misnomers that fibroid symptoms become a non-issue during perimenopause and after, and we know that not to be the case. So can you speak to some of your insights as women with a higher estrogen load approach perimenopause and menopause and how they can be thinking about their care as they move into those years?
Yeah, so that's a really interesting question. And I think, again, what we're seeing now around perimenopause specifically is more and more research that tells us that it's not just a case of we go into this decline of estrogen as we move into perimenopause. What we see certainly in early perimenopause is that estrogen fluctuates wildly in that first half of the menstrual cycle where you finish your period and estrogen it starts to rise. And that before perimenopause, that's a gradual rise and a peak right before ovulation. What we're seeing in that time is huge variations. And if we know that fibroids are fueled by excess estrogen, and we're seeing these huge variations of estrogen during perimenopause, we actually have to think everything that I've talked about around managing estrogen load, managing, um, supporting estrogen detoxification in the gut and the liver, that becomes even more valuable in early to mid perimenopause, where we have these huge fluctuations. Something that is also really valuable in certainly early to mid perimenopause, where we're still seeing regular ovulation is doing what you can to not only support ovulation, but that release of progesterone that happens after ovulation. Because if we think about estrogen, the two major hormones that we're talking about here, estrogen and progesterone, they work like a seesaw or a teeter-totter along the menstrual cycle. So we're looking at can we do to make sure during the second half of the menstrual cycle we're releasing as much progesterone as possible to help balance out the the estrogen and the excess estrogen that we see with fibroids. So there's a lot of, as you said, misnomers when it comes to fibroids and perimenopause. So something I've heard is that fibroids become a non-issue during perimenopause, and Sorry, my dog just barking in the background. Um, Something I've heard is that people say that fibroids become a non-issue during perimenopause, but that's really not the case. So continuing to do what you can to manage the estrogen load, estrogen detoxification, that balancing out of estrogen and progesterone via ovulation is very, very valuable.
Incredible information again. And I want to highlight too your point about the thyroid and selenium. I cannot tell you how many practitioners I talked to about thyroid symptoms. Like I felt my thyroid vibrating during the perimenopause transition, and they looked at me like I had lost my mind. Like there was no correlation, no understanding from their training about the relationship between the thyroid and perimenopause, menopause, and it was a huge issue. So thank you for pointing that out.
I
I would
just Can I just add one more point on
absolutely
that? Yeah.
sure.
There is actually something that we now call the thyroid pause, where because of these big estrogen fluctuations that we see in early perimenopause and mid perimenopause, that is having an effect on the thyroid. And so there are women who have these typically hypothyroid or underactive thyroid symptoms turning up at their doctors and saying, what's going on with me? I'm so tired. I can't think straight. And then they have their thyroid tested and they're seeing, okay, actually there's an issue going on that needs to be addressed.
Mhm. Yeah, I totally agree. I think that perhaps the markers that we currently have are not really capturing what's happening with everyone, and that's a huge gap that needs to be addressed. Um, so I'm curious to hear from each of you if we are to if we had the funding that we should have for a health concern like fibroids that impacts millions and millions and millions of women in countries across the world and of all ethnicities. Can you highlight two areas that you would like to see funding just flood into to kind of start this funding process?
Dennis, you want to go first or?
Okay, sure. So I would say the first would be more non-surgical treatments for fibroids because we still hear so commonly that women and people with fibroids being told that the only way to truly manage fibroids is through a hysterectomy. And we know that that's really not the case. So I would love to see more funding for non-surgical um non-surgical treatments. And then I love just to see more research on the cause of fibroids. Like why we need to understand what causes fibroids.
Thank you. Dr. Adli?
Yeah, I 100% agree with Lenny's. To be honest, removing the hysterectomies, which, in the US alone, 300,000 hysterectomies are being performed, and this is not the solution. And we are removing the entire uterus during this surgical procedures. And uterus is not just an organ; it's also the organ where the tumor develops. It's the organ that produces all this woman hormones. By removing it, it's not really, should not be even an option. But I think right now that we don't have any other options, and that seems to be an option for us. And that's why, anything that we do, and also I believe should be a non-hormonal treatment, because, by just dumping tons of hormones to the body and making body go into a menopause in a way is not also a solution for these treatments. That's why we are very excited about finding this kind of precision medicine that will selectively kill these mutant cells. It's a non-hormonal pill. That's our purpose. That's kind of our mission, the mission of me personally, and also the Rosa Therapeutics to develop this kind of molecules that we can selectively target these mutant cells. I think up until now, we couldn't even imagine doing this kind of screening. We need to perform high throughput unbiased screening to be able to identify these kind of molecules. And I think with this, now that we are able to develop model systems for this, we can actually screen for these kind of things. And not only chemical screening, also we can now perform whole genome, what we call genetic screenings, where we have a mutation, and then we are saying that let's knock out all in these model systems. Let's also delete one gene at a time, entire other genes, to identify this selective synthetic lethality targets. So this term, this synthetic lethality, we borrow from the yeast genetic model system works, is basically is saying that if you have one abnormality, can you do something else that together with this abnormality will now kill these cells? And we have a very successful examples of this in the cancer treatment. For example, we all know BCR mutation, BRCA mutations in ovarian cancer, for example, that creates cells certain vulnerability, and now we can treat these BRCA mutations, patients that have these BRCA mutations with PARP inhibitors, which is something completely independent of this mutation. But together, if those patients that have BRCA mutations, if we give them PARP inhibitors, their tumors shrink and go away significantly. And we want to achieve something similar in fibroid treatments, that you may have a MET12 mutation, but we are identifying now through this genetic screening that if you inhibit something else, now together with this MET12 mutation, they become synthetic lethal combination. And one of the goals for us, I think, is that we are invested in, and we are seeking more funding in it to identify such synthetic lethal combinations for fibroid treatments.
This has been just such a fascinating conversation. I have a lot more questions, but I want to be respectful of our time, and I am inviting you all to come back and speak with us again and keep us updated on your work, because this conversation that we're having today is directly correlates to something that I say often, which is that you need a team. And that team is a balance between the science and medical and the holistic and the nutritional. And this conversation is evidence of how those two spheres are necessary. We need to merge them, and they are critical to the forward movement and the understanding of uterine fibroids, in my opinion. And I can't thank you both enough for providing these significant insights today. I'd like to give you an opportunity to let everyone know where they can find you. Lanice, where can our listeners find you?
So my website is www.eatlovemove.com. And you can find me at Eat Love Move on all social media. My podcast is Period Story, and it's available anywhere you find podcasts. And then my book is You Can Have a Better Period, and you can get that anywhere you buy books online.
Thank you. And we will provide the links when this is aired. And Dr. Adli, where can our listeners find you?
Yeah. First of all, thank you so much for bringing us together and providing this platform for us. I'm a professor at Northwestern University. My lab website, is adli.adlilab.org. My email address is adli@northwestern.edu. And also listeners just can type my name into Google. I think they will go find the website either through Northwestern or through the lab website. It has been a pleasure to join you.
And the pleasure is ours. Thank you so much. This is this far exceeded our goals for this conversation today. And we fully intend to invite you back and build on this really, really pivotal conversation that has revealed so much. So thank you both. Happy Fibroid Awareness Month, and please continue what you're doing. It's awesome.
Thank you.
This concludes our programming that was funded by the Patient-Centered Outcomes Research Institute for Fibroid Awareness Month 2026. We'd like to thank PCORI for their support of our patient-centered advocacy efforts, and we'd like to thank you for joining us and continuing to support this community. We look forward to sharing future programming and events with you in person and virtually. Please support the Fibroid Foundation. The link can be found at fibroidfoundation.org. And please let us know how we can continue to support you in your fibroid journey. Together, we are making a difference. Thank you.